I'm going to add to the land acknowledgements with the fact that most of my work's in Central America and is supported by people who still identify as Maya or descendants of the Maya. And in fact, it's Mayan structures that make a certain amount of my own work possible. So what I want to talk about is the very thing that almost nobody else in the room will know much about, which is DNA in the air. Most of us think of eDNA as an aquatic science, and that is indeed the vast majority of where the research has been. This is one of my students, Alejandro. He's trying to find last remnant populations of axolotls in the Mexican Rift Lakes around Mexico City. And this is where we think of eDNA as being a factor in aquatic systems, and it's best known where that's where most of the work has been. But eDNA actually began on land. The first things that we properly called environmental DNA were terrestrial, and in fact, on land, there are vastly more potential sources for eDNA other than just water. Honey, soils, rain, the blood meals of blood-feeding insects, footprints in the snow, more recently, swabbing leaves, the surfaces of insects, oops, jumped ahead. Even charcoal have been found as really good sources of environmental DNA on land. And one of the problems on land is there's so many sources, we don't have a source. No one's quite sure what to use. In fact, the most widely used terrestrial source for environmental DNA these days is probably secondary sources, like blood feeding or interacting insects and invertebrates. So my students have used things like leeches for the blood meal, and from leeches and blood meals, or from dung beetles rolling mammal dung around the forest floor, you can actually survey tropical diversity very well. This is work done in Borneo as part of the SAFE program in ecology. And I sent Rosie Drinkwater off to try and find mammals by finding leeches and dung beetles, and she came back from DNA from things like pangolins and banded civets, some of the most trafficked and endangered species in the world. Had I sent her off to find me a pangolin, she would have probably failed. I sent her off to find leeches, and she found pangolins. So these secondary samplers are some of our best tools, because they are like a little army of field assistants interacting with other things in the environment that are really hard for us to get to. There is a lot of aquatic work on terrestrial diversity. I pulled this from a paper that was published 2 years ago, and it compares all the different techniques for sampling mammals, from things like, what have we got? We've got pitfall traps, grid sampling, mist nets. If you're a bat person like me, you use mist nets, camera trapping, probably the most common mammal sampling technique. All of them pale in comparison to water. You just go and pick some water out of a local river, you can get more representation of mammal diversity from the water than any of our existing techniques on land. And that's either a really good vote of confidence for water or a pitiful vote of confidence for our mammal sampling techniques. But none of them recover as much diversity in one go as water does. And it was in the context of this about 3 years ago that I was commissioned by a branch of the Chief Scientist Group of the UK where I was working to write a report on how we should use DNA as a protective measure of ecosystems on land. They commissioned 5 reports from 5 different academic or industrial groups, and the UK is very forward thinking about how to use DNA in ecosystem resilience, protection, and conservation legislation. This is part of their 25-year plan. How do we use DNA on land to cover ecosystem resilience? They commissioned one on water, one on bioinformatics. These were all supposed to be speculative, future thinking, what could we do next kind of documents. They called them think pieces. So I was asked to write the one on land, and I wrote all about different sources of DNA on land. I wrote about soils and honey and leeches and dung beetles and all the things that I knew about, and I wrote air. And then when you write a government report, you have to go find case studies to back up why this might work. And I found one for everything except air. I just assumed someone had looked. All I could find at the time was a lot of papers that sort of said we should look in air as a theoretical thing, a small amount of work on plants and fungi. And mostly this was in the context of things like pathogens, allergens. We had a lot of knowledge of microbes in air. We've known that for a long time. 20 years ago, went out sampling with somebody who was collecting viruses from the air. And in a COVID world, we know this. But no one had really looked for anything other than the pathogen. a tiny 1 or 2 papers had mentioned plants and fungi. And so I wrote, we should look in the air like everybody else. And then coincidentally, the same month, my own university, Queen Mary, offered some funding for what they called high-risk, high-reward science. Basically, give us a crazy idea, we might fund it. And they awarded me a small amount of funding because I told them I wanted to vacuum the sky. They don't just award you funding for that. First they say why, and then they say how, and then you say things like, well, I've been kind of inventing techniques for doing this stuff for years, and here's what we think might work, and I've got this perfect setup, and they say, oh, okay, we'll give you some money. So they gave us some money, and the first thing we did was we went to our own lab, where we had colonies of naked mole rats. And our thought was, if you're ever going to demonstrate that DNA is carried in the air, we should start with something that lives in high density in a small confined space for a very long period of time. Because if DNA is ever going to build up, we'll start there. So we took some samples from the boxes and the artificial burrows that they live in. These are eusocial colonial mammals. They've got a lot of other cool things about them, but naked mole rats are great. And then we took samples from the room, assuming that would be our negative control. So our expectation was mole rat DNA in the mole rat burrow, no mole rat DNA in the room. And what we found was DNA absolutely everywhere. Mole rat DNA, people DNA, dog DNA that someone had tracked in from when he walks his mother's dog on the weekend. Secondary transfer of forensic tiny amounts of DNA leaching into what should be a closed facility. There was DNA everywhere. We didn't have any negative controls. Everything had DNA in it. And so we thought, well, maybe this is a little easier than we thought it was going to be. And so the next thing they say, what are you going to do to prove you can use this? I said, we're going to go to the zoo. So this is, seems to skip a bit. This is Hamerton Zoo Park in rural England. This is when COVID hit. And so actually this was within my allowable travel distance legally at the time. So I could go to the zoo. And I went to the zoo with my field assistant. And we walked around and we took air samples all over the zoo. And we took air samples like on the edge of their enclosures where we thought, kind of like the mole rats, if they're going to live there, there's the dingoes looking at my sampler. That's where DNA should be. But we also took samples outside where there were no animals, just open area. And the reason zoos are wonderful places is the same reason that fish tanks were the original good place to look for aquatic eDNA. You take an environment, you introduce a species that shouldn't be there. If I'm in rural England and I pick up cow, there's no way of knowing where that cow came from. But if I pick up tiger, there aren't that many sources in England for tigers. So that was the idea of the zoo. If we walk around and we take samples of what are basically a colony of invasive species, can we detect them outside when the atmosphere is really big and the animals are really small? The answer was yes. So this is a top-down view of the zoo. And here are 7 enclosures where we tested stuff. And here are the things in the enclosure being detected by DNA. So in the Tyra enclosure, we detected the Tyras, which is very good. Over in the primate house, we detected a bunch of primates. And that's kind of what you hope you're going to do. But more interesting than that was the other stuff we detected. So in the Tyra enclosure, we also find lemurs and binturongs and dingoes and budgies. This is DNA moving around the zoo. This is way, way better than detecting a Tyra. Because this, if I have to be right on top of the Tyra to detect a Tyra, there's no point in sucking DNA around a Tyra, because it's right there. But if I can be hundreds of meters away from it and detect it, then it's a useful tool, just like in the aquatic systems. We also found native wildlife, squirrels, hedgehogs, ducks, Myotis bats. Hedgehogs are really cool because they're hugely protected in the UK. Muntjac deer are locally invasive. They're released by game parks of the royal families and the estate owners. And they've become invasive, although they're a Southeast Asian deer, they're big invasive species in the UK. This is wildlife basically floating into the zoo, even further away. And then we detected the stuff they ate. And I started off as a dietary ecologist, so I really like the idea that we can detect predation effectively. If you're in the Tyra enclosure, you detect a lot of chicken. That's their favorite food. But if you move over to the dingoes, it's mostly pig, horse, and cow, because they prefer those to chicken. So we're finding spikes of their favorite prey in and around where they live, which makes sense. This is probably aerosolized feces. At the same time, over in Texas, a group was working on passive sampling. This is a dust trap, and they set out these dust traps to actually catch plant material And they sampled them every other week for an entire year to see whether they could pick up plant material in the air. And yes, pollen is part of that, but many of these things were not things that have wind-borne pollination they were looking for. So is there truly plant material in the air beyond the obvious pollens that anybody who has hay fever knows about? And the answer there was quite yes. And they published several interesting papers, and around the same time we were working with mole rats. And then they called us and said, you know, we looked for plant material, but we never looked for anything else. Would you like to retest our DNA samples for animals? So we said yes. And we found them. This is passively collected dust from the air in a rangeland. And this is each of the 22 samples across a whole year, so every other week. These are the animals, and these are just the green boxes are detections. And there's a couple of really important patterns that tell us about airborne DNA. First of all, common things like bunny rabbits in Texas, you get them almost every week. So common stuff is commonly detected. In contrast, rare stuff is rarely detected. This is a very strange little toad, it'll come back in a minute. It was only detected once, it's very rare. We also have a big blank space where there's not much detected. This is actually a good thing to find, because this is the time of the year when it's hot, the vegetation goes away, most of the animals go away. So in a time of year where we expect low activity in this landscape, we get very little detection other than bunny rabbits, which pairs up with what they know is happening in the landscape. In contrast, this is cooler, rainy, windy mating season for Texas. And the animals get active, and they come back, and suddenly we begin detecting them. And the most important one is that frog, or that toad again, which is an ephemeral toad, and it only really emerges in the dry Texas landscape when you have an aquatic event. There was a huge thunderstorm 1 week before we detected it. It probably rehydrated a pond on site. These little ephemeral toads come out, they quickly mate, and they go away again. You never detect them again. So this suggests that like aquatic eDNA, it's highly heterogeneous, everything is not everywhere, and that like aquatic eDNA, the detections correspond to activity, and that's what we want. If animals are turning up, and we then detect them, this is a good thing. If we detect them when they're never there, then it's not a useful tool. So with this sort of idea that this was something we could actually track activity in a certain regularity, we set about building our own little samplers. So these are my kids, and my nieces and my nephews. That's my daughter, that's my son. He's given the Play-Doh, he doesn't build stuff. But they are building handheld samplers on my table based on the design that we modified from Denmark. We have a new prototype, which we are testing this Wednesday for the first time. But they basically look like this. They are a fan from a computer with a 3D housing and a super secret material as a filter, which all of you stick in your furnaces every day. So furnace filter material is rated to catch viruses, and therefore it catches DNA. So furnace filter material, which we take apart furnaces from the filters from wherever you want to go, Lowe's or Home Depot, we cut out the little pieces, sterilize them, and stick them on filters. And we use these, we power these. Here's my nieces and nephews. If you live at my house, you have to spend your weekends out doing science. So March break, I dragged all my nieces and nephews out to help me test these things. They're looking at bird boxes there. We took them down first to Central America, where we work. I'm part of a team that makes and has done an annual survey for bat diversity for going on 17 years now. It takes 60 of us 2 weeks to do a good survey for bat diversity. One of the things that happens is these bags has a bat in it. This is how you hold a bat in captivity briefly. They come into a room, and my job is to identify them. That's what I'm good at. That's what I spent my time here at Guelph learning how to do. So I can identify all the bats. So they all get lined up on this string, and I have to look at every single one and confirm the identification. Notice in the background, the air sampler is deployed on the here. So the question we had was simply, let's pretend this is an artificial cave, and the animals are coming and going every night for a few hours over the course of weeks. If we had just placed samplers in a cave, where this tremendously complicated community is coming and going, could we have described the diversity of that quote-unquote cave? And the answer was yes. In fact, we were 90% accurate at describing that room's diversity. There were 40 species of bat, roughly 1,000 individuals. Some of them were only one, some where we had hundreds, and we were about 90% accurate at telling you what species had been in that room over 2 weeks. And a lot of other stuff, which we learned about DNA floating through windows. We have gone back. The next project we worked on is called Species at Risk Monitoring, basically, where we have deployed these samplers inside natural roosts like this one. This is actually a tunnel carved into a Mayan structure. And if you want to hear about species at risk monitoring, actual applied science, my student Nina will give a talk on that later this week. That's Nina in a cave. We're also working on smaller things. We recently were funded to study how we could use airborne eDNA to study bees, and also to ask questions about the ethics of eDNA. Because it turns out this wonderful non-invasive technology has questions attached to it. A really good one being, if it's really easy for you to collect the DNA, anybody can collect it, like poachers. So who has access? Who owns it? So there's actually a bioethicist here at University of Guelph who is evaluating the technology. Is non-invasive technology actually ethically superior, and why or why not? Well, we develop it. So we're trying to take that approach. And so this is what we're doing. We have solitary bee homes, we have bee hives, and we are trying to study the use of eDNA in small diagnostics, bee DNA. So what we've learned about airborne DNA so far is that, yes, animal and plant DNA is carried in air. It works just like water. Virtually every rule you know about aquatic eDNA probably applies on land. Air currents versus water currents, it's all the same. It can be passively or actively collected. Both work really well. It appears to reflect recent activity, and it can accurately describe highly complicated tropical communities. We can do that with something like a bat group. Now, the problem with all of these, and it's not an eDNA problem, it's a biodiversity problem, is that every technique we have is really based around small-scale intensive monitoring. The bats in the cave, the frogs in the pond, whatever it is you happen to look at, you're probably targeting one river or one lake or one pond at a time. That is not what biodiversity needs. None of those can tell me what lives here. And this is actually my first field site from when I was a grad student here at Guelph. This is Guanacaste in Costa Rica. Now, the first thing about this is now that I see this mist rising off, what I used to think was really nice mist, I now see, oh, DNA. Probably being coughed out by forests every single night. And I have some big plans about new forms of quote-unquote mist nets. It may involve me having to fly in ultralight, I don't know. But how do you measure this? Like, how do you understand a landscape with any biodiversity technique we have? There isn't anything that scales. Other sciences scale. The satellites and the astronomy people, they can measure what they need to measure across the whole planet or a whole galaxy all at once. We can't do it. We still work on bioblitzes. And this is not a scalable method. Biodiversity has three problems. We have no standardized method. Even in the aquatic eDNA, we don't standardize anything. And because we haven't got standardized methods, they're all taxon-specific or habitat-specific or technology-specific. We have no standardized metrics. Well, some basic ones like a diversity index, but nothing that really scales with good statistics. And the reason for that is that we don't have any global infrastructure. Other sciences do. We have massive infrastructures for other disciplines, physics, astronomy. They figured it out. They can do this. So can we. Last January, a physicist called me and said, I saw an article on the Natural History Museum website about air sampling. What are you doing? And I tried to explain biodiversity to the physicist. And he said, what do you mean you haven't got any standardized methods of measuring things? This is a physicist, right? It's like, what do you mean your science doesn't standardize? There's a science that doesn't standardize? So I explained the problem. And he said, so all you want to do is measure air? Like filter air? I said, yeah. He said, and you're building little devices? I said, yeah. He said, have you thought about air quality control centers? I said, what are those? He said, well, I operate them. And this gentleman was named James Allerton, and he and his friend Andrew Brown, who do that wonderful thing we tell students to do, they read outside their discipline, had picked up on the biodiversity story about aerosols. And these are metrologists. They have globally deployed, highly standardized air sampling equipment. This is one of them. This is a head that sucks air in a precisely 1 cubic meter per unit time for exactly 7 days. It then automatically switches to a new filter. It does it again and again every single week all year long. And they are looking, in this case, this is the heavy metals network, and they are looking for things like offshoots of the petroleum industry, lead, arsenic, manganese, things that are part of human health surveillance. And they filter them onto these little discs. This is a 47 millimeter disc, and by the end of filtration, it looks like this. So it starts like that, comes out like that, full of stuff. He said, is it possible we're sampling eDNA at the same time? This is a smaller pore size than anything you've ever used if you work in eDNA. So what's on it? And they sent us some of these little quarters. They take a quarter, they analyze it. They take another quarter and they put it away for long-term storage, and then they throw it out. And it turns out everybody throws it out. Canada throws it out every single day. We have daily samples that are thrown away. So we set up an experiment with them. We went to the Scottish heathlands. This is what Scotland looks like. Big empty area where they happen to have these samplers set up because, as people keep saying, isn't this a city thing? No, actually they take background measurements. They have to know how cities compare to the background environments. They take background measurements everywhere. This is Scottish, outside Edinburgh, about 20 miles, in an area whose name I can't pronounce, but they take measurements here. So they took a bunch of those and they gave it to us. This is part of their network. So these are just regular operating networks. They have their own machine, because it turns out most of these things are actually private. They're not part of national networks. This is outside a place called Bushy Park in London. So one of the biggest urban centers in Europe. It has an urban forest. Bushy Park is big enough to have herds of deer running around it. It's an old game park from the royal family. And in Bushy Park, it was not part of the network, so we could manipulate it. We took samples for an hour, a day, and a week, and then we did it again, and then we did it again. And then they sent us some from Scotland, and at the time, we did not know that the ones they sent us from Scotland were nearly a year old. They had sat at room temperature for a year. And to cut the chase, we couldn't tell the difference. 2 locations, 17 samples, 180 different taxonomic groups simultaneously recorded on filters. 80 different genera of plants. In London, you get ornamentals from gardens. You get crops from the agricultural fields. In Scotland, you get heath plants, which you expect. 36 different species of bird, 24, something like that, different mammals, fungi, a couple of amphibians, 6 orders of insect. I think there were 8 different phyla covered in this sample, all simultaneously recorded by a system that already exists. And the samples that come off the machines simply go straight into a DNA extraction tube. No changes need to happen. It doesn't even disrupt the operation of the network because the stuff from Scotland worked just fine. And in fact, what we did recovered in our different sort of experiments is the only graph I'm going to show you, makes total sense. So here down in London, where we've had 1 hour, 1 day, 1 week. After an hour, you haven't picked up very much. You're actually getting quite a few plants because plants don't move. They're there already. But you don't get many insects or animals because they haven't moved in yet. If you wait for a day, you begin to pick up insects and some mammals. The plants peak. So after a day, you've pretty much saturated on plants because no new ones are going to move in over the course of your experiment. But if you wait a little longer, the mammals and the birds will get higher because after a week, this is based on encounter frequency. These things move. They come and go. So the longer you wait, the more things are going to come into the area and get picked up by the filters. Out in Scotland, very comparable data. Look at the plants in particular. In fact, these Scottish samples, which operate for a week, are indistinguishable from the week-long fresh ones from London in terms of number of plants you picked up. So it works. A little less because it's not London. You haven't got the ornamentals. But the natural plants are there. We do have a drop-off in insects, which we have actually solved. This was a primary issue, I think. But the vertebrates, we're getting comparable vertebrates after almost a year of sitting at room temperature with no DNA preservation in mind. So the mammal and bird DNA was intact. So now we have 3 different priorities in biodiversity science. How far does this stuff go? We have no idea. I mean, I can't tell you how far this DNA is being drawn from, but the metrologists can, because they have spent the last 80 years designing these systems with precise knowledge of how far particulates move under certain circumstances. They know everything about this. And they can give us models of spatial effect as our baseline hypotheses. They've already worked it out. They've designed these systems to cover as much land as they can in the perfect scenarios. And so we have these baseline estimates that we can use For DNA, we need to understand what the temporal persistence of the signal is. And within environmental DNA work, we hold two competing hypotheses. One is it's incredibly fragile and it degrades, coupled with it can hang around for hundreds of thousands of years, somewhere in the middle. But how long do you have to sample? How much do you have to sample to actually measure using these systems? And how well does it match known distributions? And it turns out in the UK alone, there are 150 sites taking weekly samples. That is 8,000 absolutely identical samples collected in just the UK, Scotland, and Northern Ireland. We've gone from having not enough biodiversity data to more than I can handle. 8,000. They've been measuring since the 60s. Some countries have saved them. Most haven't, but some have. There are archives going back 70 years for some of these. And there are thousands of these. In the UK, we have the perfect scenario to test these hypotheses. This is just the heavy metal network. There's the site we worked at near Edinburgh. This is all the other sites that we can get samples from. And the reason that the UK is really, really good for this is they are uniquely good at knowing where their biodiversity is. These are maps produced by a citizen group that surveys deer. This is absolute abundance data to the square kilometer for every deer species they have. They have similar data for all kinds of different key taxonomic groups. They know all the forests, they know the arable crops, they know when they change. There is annual Christmas Day bird counts. There's the butterfly people who go out on set days every year and measure butterflies. There's the Spring Keeper Frog Watch. There's the Migratory Bird Society. They keep incredible citizen science records of when things come and go, which gives us the data we need to actually figure out what it is we're measuring and how close it might be to one of these maps. And then we started thinking, well, how much further do these networks go? This is Europe. Because, of course, the UK was recently part of Europe. Europe is governed by exactly the same requirement law. Every one of these sites takes exactly the same measurement that England does, in the same way, because they're governed by the same transnational law. And it's all about keeping particulates for human health below a certain level. But as a byproduct, they are measuring biodiversity at tens of thousands of positions every single day. This is the States. They have 3,000 sampling stations. About 1,000 of them are collecting the samples. We have been doing this since the late 60s and early 70s. And the next question people ask is, well, is this just industrialized countries? Nope. We have found them in the polar regions. We found them in the Amazon. They're everywhere. This is not an accurate map. This is just citizen-reported data of samplers that measure in the right size range to match our data. I physically filtered it. This is just citizen-reported stuff. This is not national networks. I don't even know if they save a sample. These might be real-time measurements that don't actually save on a filter. It's impossible to actually get a map of the planet this way because, in fact, the planet, most of it is private. If you turn your phone on and switch your weather app on and scroll down to the air quality index, you are looking at a private network measuring the same thing. So this is how you measure biodiversity at scale. This is a standardized, repeatable, highly, highly nationally regulated and controlled system which is already taking biodiversity measurements. We've shown it works. I don't know how to deal with the sample size. I've never had that problem in 25 years of biodiversity science of not knowing what to do with the data. There are literally millions of samples in existence like this. And if they're viable, it is a historical record of biodiversity going back decades. Thank you.